Okay, so to kind of close out this session, I just want to talk about some work that we've been doing to understand how rTMS, specifically subthreshold rTMS, drives plasticity in the brain. So when we think about how rTMS drives plasticity, most of us will think that rTMS is driving synaptic plasticity. And I guess one of the major lines of evidence to support this is that in humans, if you give rTMS and then you give them a blocker for synaptic plasticity, a NMDA receptor antagonist, you can kind of block this rTMS-induced plasticity. So this definitely suggests that synaptic plasticity is being induced, but unfortunately, there are no direct ways of measuring synaptic plasticity in the human brain because it's very invasive. And so that's why we need animal or experimental models. And so I think one of the best examples of this is the study done by Andreas Vlachos, done a couple of years ago now. But basically, this is where they take rodent brain tissue. I'll just get my pointer. You take rodent brain tissue, you grow up in a dish, and then you can deliver rTMS to it externally. And what they found is that if they measure the synaptic strength of excitatory neurons directly, they saw an increase in the synaptic strength of excitatory neurons. Interestingly, this only occurred about 2 to 4 hours after stimulation, which I can't go into now, but it's a bit different to what you see in humans and MEPs. But in addition to synaptic strength changes, they found structural changes. And so here we have the dendritic spines. These little knobbly things here are the dendritic spines, the postsynaptic structures. And what they found is that in addition to their functional changes, that the size, in this case, of the synapse changed. And so this is in line with other animal studies of synaptic plasticity, where generally a functional change or a change in synaptic weight will always have, or almost always have, a change in structure that follows with it. So this is suggesting that rTMS is also driving structural plasticity in the brain, but there's currently no evidence to suggest this happens in a living brain. It happens in vitro, but does it happen in a living brain? And so to do this, we turn to rodents. And one of the powerful things about rodents is that you can take advantage of the genetic models that are available. So this is a mouse that's been genetically modified to express green fluorescent protein in a subset of excitatory neurons, as shown here. And so using this technique, what we can do is actually replace part of the skull with a glass cover slip. And this then allows us to actually image the brain directly. And so doing this, we can actually see the living synapses in a brain. And we can come back to these synapses every day and see how they change over time. So this is an example of some of the images we've taken. So this is an example of multiple dendrites in the field. This is kind of like a bird's eye view of the brain. And you can start to see here some little knobbly things here. These are dendritic spines. And so we can come back to these spines every day and see how they change with rTMS. One of the things that we're looking for are changes in dynamics. So what we can look at here are dendritic spines. We can see which are the spines which are stable, shown in the white arrows, which are the spines which disappear over the time, which are shown here in the red arrows, and which are the spines which actually grow, come into life over the course of the experiment, shown here in green. And so using this technique, we asked ourselves, does rTMS induce structural synaptic plasticity in the living brain? And so we did 2 experiments. This is the first experiment. So after we put our cranial window into the mice, we can do baseline imaging to get an idea of how the dynamics change normally and the density before rTMS. We can then give our rTMS in the form of subthreshold ITBS using some rodent coils I made with Jenny Roger during my PhD. And then we can look at how rTMS has changed our spines post-stimulation. And we do this in 2 groups. We have a young adult group, which is 6 months old, and we have an aged group, which is 2 years old. And that's very old for a mouse. And we do this in the motor cortex. Our second experiment was to look at, well, perhaps multiple days or multiple sessions of stimulation is driving more plasticity than a single stimulation. So to do this, we do the same. We put our window into our mice, but this time we give 4 daily sessions of ITBS along with daily imaging, and then we follow it up again with post-stimulation imaging. But it's important to realize this is only done in the young adult mice. We don't have an aged cohort here. So I'll go through the results. So looking at our single session of ITBS study in the young and aged mice. So we look at spine density here. Here we've plotted both ages on the same plot. We've done a Bayesian analysis to look at evidence for changes. So in blue here, this is our pre-stimulation time point. The thick line represents the mean of that time. And then we have our plus 21 hours and our plus 45 hours post-stimulation. So we ran our Bayesian analysis to look for evidence of change. What we found was that there was a strong evidence for a decrease in spine density at the plus 45 hours post-stimulation. Now, the data shown here is spines per micrometer. So when you extrapolate that over, say, larger areas, so say 100 microns, this results in a change of about 10 less spines, so 10 less synapses per 100 microns of a neuron. And this change was present in both the aged and young animals. So rTMS is driving the structural plasticity irrespective of the age of the motor cortex. When we look at spine losses, what we're seeing is that there was strong evidence for an increase in spine losses at the plus 21 hours post-stimulation. And again, when you extrapolate this over to about 100 spines, this is an increased loss of about 4 spines for every 100 spines. And again, this didn't change whether it was the younger or aged animal, so it happened to both cohorts. When we look at spine gains, so those gains of new spines, we see that at the plus 45-hour stimulation, there was extremely strong evidence for a decrease in spine gains. And when you extrapolate this, it's about 1 less spine gain per 100 microns. And this happened in both the aged and the young adult mice. When we looked at our multiple stimulations to see whether more stimulation was driving more synaptic plasticity, when we looked at spine density, we didn't actually see any change in spine density after ITBS relative to baseline. When we looked at spine losses, we saw that there was an increase in spine losses at the plus 21 hours post each stimulation day. And that this equates to about an increase in 4 spine losses for every 100 spines, just like it was in the single stimulation. When we looked at spine gains, we found that there was no change following ITBS, which was different to the single stimulation group. So to wrap up a very short talk, this is kind of like a table of all our results if you're interested in, but basically what we found was that a single session of subthreshold ITBS was having a long-lasting effect on structural synaptic plasticity in the motor cortex. And interestingly, this happened in both the young adult and the aged brain. When we looked at multiple stimulations in the young adult group, we found that multiple stimulations was able to maintain the acute effect of a single stimulation in that there was a change at plus 21 hours, but we didn't see any change at the plus 45 hours. And so surprisingly, the effects of multiple stimulation are not as long lasting as a single stimulation. I do want to point out that these changes seem quite small, and to an extent they are, but we have to remember that neurons have multiple dendrites that span hundreds of microns. And if these changes are occurring to hundreds to thousands of neurons in the motor cortex, this will significantly change the connectivity that is occurring in the motor cortex. So we hope to expand this to other protocols to find out how rTMS is driving plasticity. And that's all I have. Thank you.
Great, thanks, Alex. Questions for Alex. I've got a quick question, Alex. So I noticed that the variability in your results is quite large, and thanks for showing the actual results, because it's nice to see that. I see the same thing in humans, and it's frustrating because I wish we could narrow down that variability that we see. But can you just tell me a little bit about the technique? Does the mouse stay in the device for the whole 4 days, for instance, or does he around in between times and go back in? Could that be a source of variability? Or
okay, yeah. So with the protocol, so like the mouse is lightly anesthetized when we do the imaging, which lasts about an hour. The TMS itself is the standard 600 pulses, so it's stimulated awake. So it's about 3 minutes, and then when it's not being imaged or it's not being TMS'd, it's back in its home cage with its litter mates. So it's not in the TMS device or the microscope for the full 4 days. It is a good point about variability, which I think is important to realize. Like one of the most common questions I get is like, do animals show variability? And they do. So what we were showing there were actually data from dendrites from multiple animals. So what we're seeing is that we're seeing changes across the animals, but not every dendrite is responding. So I guess in some ways that's a good thing in my mind, that you're not changing every single dendrite, but it could also explain, some maybe, like obviously extrapolating here, why we see variability in humans, because rTMS is not changing every dendrite, it's not changing every neuron.
And sometimes, of course, that variability can lead to really kind of unique findings, why some people or animals respond and why others don't as well. So sometimes it's worth exploring that variability more. Thanks, great talk. Where to next?
With the experiments?
With your work, yeah.
So you've already heard from my postdoc and my student. We're looking at axonal plasticity. That's something that I'm really excited about. But in terms of synaptic plasticity, we are looking, and I'll have a symposium on this later in the week. We're looking at trying to understand how, say, like axonal plasticity feeds into synaptic plasticity, and also how rTMS is regulating, say, homeostatic or metaplasticity. We're doing that with Cliff Abraham, who came up with metaplasticity. So we're pretty excited to explore these kind of things in animals, which obviously we can't explore in humans.
Great, good luck. Thanks everybody for your time today. That wraps up our abstract session this morning.